very pleased today to introduce to you Debashish Bhattacharya, and he received both his bachelor's and master's at Dalhousie, his PhD at Simon Fraser, so he's a product of Canada. Upon completing his degree, you wouldn't expect it looking at him. After he completed his degree, he was a Sloan Fellow with Mitch Sogan at the MBL in RedToL. Unfortunately. And from there, he went, he was a Humboldt Scholar at the University of Cologne in Germany. And then from there, he went to Göttingen as a research scientist at the Max Planck Institute for biophysical chemistry. In 1999, he joined the faculty of the University of Iowa, where he was until 2009, where he joined the faculty of RUTGERS, which is where he is now in Ecology and Evolution. So, without further ado, Debashish.
You would clap your American brethren, so I'm sure. We're amongst friends here. Thanks, Gordon. So I gave a talk at Ecology and Evolution at Stony Brook in March. So this is my third trip. My daughter was here for a volleyball tournament, so I know your city just a little bit. So I was asked to talk about gene transfer and so on. So my lab is a number of different things, but I was going to focus this talk on how genomics helps us understand the flow of information amongst algal eukaryotes and how interesting and complicated that can be. The major topics of my seminar are shown here. First, I'll introduce you to endosymbiosis, which is one of the fundamental properties that define eukaryotes, the origin of organelles. I'll talk a little bit about the gene transfer from our recently completed draft genome of the red algal Porphyridium. Talk about a new model that's a very old organism described in 1895, but a new model for endosymbiosis called organelle, and I'll end a little bit of work on single-cell genomics. That's a cartoon image of Cyanophora prepared by my wife Susanna, and this picture's gone all over the world, and it's really, I thought it was a really beautiful image. So I usually start by mentioning why the kind of work we do is impossible. It would be unfair to do otherwise. This is a cute wheel of life from Wikipedia. I'm sure some of you have probably seen it, studying for exams, as it were. And all I want to point out with this is that some of the major events in evolution, snowball and the great oxygenation event and so on, especially the origin of photosynthesis, which we worked on in terms of dating it and put it in perspective as about 1.5 billion-year-old process. In fact, if you look at the very top end of it, where we are, what you'll notice is that the entire wheel is mysterious to us. That is, we're not talking about generating mutations in E. coli or yeast, where we can have the progenitor and the descendant strains. We're talking about something we or no human has ever seen and will ever see. So the entire world of deep molecular evolution is really a guessing game. It's a detective game, and therefore it has the weaknesses of not having the model in your hand. The other part of this detective game that I think needs to be stressed is that the tree of life is a tree of survivors. This is a very famous tree from Norman Pace, based on 6-units rRNA, showing the 3 domains of life. I think a more accurate picture is from my friend and colleague Peter Gogarten at Connecticut, which essentially shows that the tree of life is nested within millions of extinct species. That's cool. You can say, well, the coalescent theory will predict that we can collapse extant lineages into more ancestral stem lineages and so on. The problem is, of course, that genes move not only between living lineages in the past and present, but they actually have moved between extinct lineages to living lineages. And therefore, we have a very imperfect view of the origin of genes and the distribution of the tree of life. As much as some people would like to argue otherwise, it's a very, very big and interesting problem. So the way that my lab approaches this in terms of algal genomics and molecular evolution is through the establishment of a sequencing and computational facility. When I moved to RUTGERS, We purchased from my lab a GA, which is a Lumina machine, and recently a mRNAseq, which is a really cool desktop sequencer that has lots of really nice capacities. So we were able to do all the genomics in-house in my lab, and we also purchased servers that allow lots of distributed work, as well as high memory servers that allow assembly and so on. So all of the work I'll talk about was done in-house, and it was the bioinformatic pipelines and so on, one of which is shown here. The famous phylogenetic pipeline that I spoke about with Vassam's group comes from that, but the people who make all this work are the folks who do bioinformatics in my lab, Dana, Sana, Mihir, Shu, Udi, Rajat, and Arik. They work on different aspects. So we try to get as much of this done in-house so we can have control over the process. All right, so the kind of things that's going on are diverse, and that's really nice because genomics, yes, of course, when you sequence a sea slug versus sequence a single cell or planting, they're very different organisms, but they all have very sort of understandable genome properties, the same nucleotides, similar sort of gene discovery and assembly characteristics in many ways. So we learn a lot about genomics by working in a lot of different organisms, and that has actually been very important for us to recognize that there are certain things that we find that unify these genomes, other things that make them very different from each other, and that's the most exciting part about doing comparative genomics. Okay, so the story of algal evolution would be real simple if it was a simple story, but unfortunately, as more data and knowledge has come out, we know that algal evolution is really complicated. And it's complicated due to the process of endosymbiosis in large part. Endosymbiosis can be thought of as the most important driver of horizontal gene transfer that we have in eukaryotes. And I'll explain that in more detail soon. So primary endosymbiosis is the initial capture, as we said, maybe about 1.5 billion years ago, of an oxygenic photosynthetic cyanobacterium, likely a biflagellate protist. And that process, the engulfment and retention of that cell, led to something called endosymbiotic gene transfer. That is the intracellular movement of genes from the captured cell to the host nucleus, and lots of other complicated tools that allow those encoded proteins to re-enter it, and lots of genes were lost. This so-called protoalga is a common ancestor for a group called the Plantae, for which we provided some strong evidence recently in the paper. And members of the Plantae, the red, green algae plants and glaucophytes, the reds and the greens were themselves engulfed in something called secondary endosymbiosis. That is, one eukaryote that is non-photosynthetic engulfs another that is, and that again puts the opportunity for lots of gene transfer. We have DNAs inside of foreign DNAs captured for long periods of time in another cell. And the dinoflagellates do something called tertiary endosymbiosis, which is engulfment of one of these secondary endosymbions by another cell. So endosymbiosis is a really, really exciting but very complicated factor in how the tree of life looks to genome biologists. And this figure produced by former postdoc Mansui Yoon shows a schematic tree of life with what are called the major, or the supergroups, the major phyla. The primary endosymbiosis here gives rise to this photosynthetic clade called the Plantae. Secondary endosymbiosis gives rise to the plastid and a whole group of organisms called chromalgulates, which may or may not be monophyletic, but many of them contain a red algal-derived plastid. This is the other second primary endosymbiosis in colonella I'll talk about later. We also have evidence in a paper we published in 2009 that these chromalgulates also have lots of genes of green algal origin. So we have a very complicated scenario for gene origin in these guys, diatoms, dinoflagellates, very important organisms. Then we have independent secondary endosymbioses in euglenids and in chlorarachniophytes. Then we have the tertiary endosymbiosis in dinoflagellates. Then we have what are believed to be horizontal transfers of plastids between chromalgulates. So that's what the tree of life really looks like. It doesn't look like a nice Darwinian sort of step, from primitive to advanced. It actually is a very complicated tree where each of these endosymbioses bring in genes. Not only the organelle, but the genes encoded on the nucleus that allow that organelle to function. The way I find an easy way to explain this is the Russian doll paradigm of plasmid endosymbiosis. Four systems 1 and 2 were assembled in the cyanobacteria, primary endosymbiosis, secondary, tertiary. The essential cyanobacterial character of those genes are maintained throughout all of these eukaryotic endosymbioses. You can find cyanobacterial-derived genes in dinoflagellates, as in red algae, as in chromalbulates, as in the plantae. So it's the transfer of large amounts of information, dozens, hundreds of genes from one lineage to another. For as long as the phyletic signatures of those genes remain, we can trace the history from even the most complex scenario back to the source. What complicates this, of course, is subsequent plastid loss, horizontal gene transfer, and divergence rates variations across genes. So all of these things make it more complicated, but this is essentially how we can explain endosymbiosis in the tree of life. So how do you study all these complicated processes? You sample the genomes, you sample the organelle genome, you look for endosymbiotic gene transfer to the nucleus, you sample the host genome, look for examples of EGT, you look for horizontal gene transfer, and you look for existence of mitochondrial genes from the mitochondrial genome that have existed there. So a typical algal genome will have at least 3 types of genes: endosymbiotic, horizontal, and mitochondrial targeted genes. And you study the machinery that allows the translation and the translocation of these proteins back to the organelle. And you also study the machines called the transporters that allow metabolite balance. So there's a number of really key machines and genomes that algae have that we can target to figure out how endosymbiosis has proceeded in such a complex arrangement. So if we were to look at this type of movement of genes through EGT, and we were to look in the group of plantae, we find that between 500 to 1,000 genes in the nucleus of red algae, green algae, and glaucophytes, in fact, originated from the cyanobacterial endosymbiont. This is a former postdoc, Adrian Reyes-Prieto, who did some of this work. So every alga is carrying around about 500 to 1,000 genes that were transferred from the nucleus, from the endosymbiont genome to the host nucleus. And there was, for a long time, there was a lot of controversy about whether or not that plantae group that I have mentioned, which was the birthplace of endosymbiosis, was in fact monophyletic. In 2 papers published by my lab, one in Current Biology and the more recent one in Science, we provided very strong evidence based on the genome of Cyanophora paradoxa, which is the glaucophyte, that there's in fact a single origin for the plantae and that the origin of the plastid happened in its common ancestor. I'm not going to talk about that paper, but if you're interested, you can have a look at it. And what it pointed out in this little article that Fred Spiegel put forth with the paper is that the common ancestor of the plantae was in fact a really complicated eukaryote. This notion that the root of the eukaryotic tree is somehow primitive organisms doesn't really hold up. If you look at the characters shared by the 3 different plantae, you find you have a pretty complex, modern-looking eukaryote minus a plastid, biflagellate, alcohol fermentation, standard sets of sugar metabolism, very, very similar to what you might find in other eukaryotes, only transformed by endosymbiosis. Okay. So let me start by talking a little bit about the Porphyridium genome, and I want to give you sort of a blow-by-blow analysis of how genomics is helping us understand the role of gene transfer in these genomes. This genome was sequenced as part of the RedToL Tree of Life grant, and initially it was meant to target 16 red algae for plastid genomes and transcriptomes. With the available of better and cheaper sequencing technologies, we've moved from ESTs and organelles to complete genomes and functional genomics. And that's allowing us to generate a really broad and robust scaffold for the tree of life for the red algae based on genome data. So I'll talk today about the Porphyridium cruentum genome project. This is a pretty famous alga if you're interested in photosynthesis. Paul Falkowski used to be around here, not here, but nearby a few years ago. He and a lot of other people actually worked on phycobilins and photosynthesis using Porphyridium as the model. So it's a very famous biochemical model for understanding photosynthesis and light capture. What's really interesting from an evolution point of view is that the existing genomes we have are from a group called the Cynidiales, Cynidus guys on the Valley area. These guys live in hot acid water and soils, Yellowstone Park and so on, they have very specialized and reduced genomes. So much of what was known about red algae was based on analysis of these genomes. Porphyridium provides a really nice model because it's a mesophile. It's found in brackish freshwater, and it has what we believe to be a normal-sized genome. It's a normal eukaryote, so it provided a really nice unicellular model to understand red algal evolution. On top of that, of course, we were very interested because it has some really important applications in industry, some of which are shown here, and potentially also as a biofuel source, not the most likely one, but certainly some interest in that as well. So what I did in this case is that I ran a course for our fuels hybrid class last year, which is it's a biofuels hybrid, and I invited all the graduate students who were part of the course to take the new genome and to annotate and analyze it on their own. So each of them took a bit of the genome and they spent a semester understanding it, and they wrote pieces of it. In fact, the great thing is I'm able to collate all that great information and put it together in the paper, which is being prepared right now. So they did a really, really fantastic job, and through it they learned how to use some genomic tools and how to think about large data sets, but asking very specific questions, for example, about the origin of sex and meiosis and so on. So here's what the data I'll talk about today looks like. We're going to have a lot more soon, but it's not going to make, I think, a big difference. 56 million reads, and about 7.5 gigabases of data. Looks like, if you add up all the contigs that are unique, it's a genome of about 20 million base pairs. So about a 20 million base pair genome, and an N50 of about 20,000, which suggests, this is the definition of N50, suggests that we have much of the genome in large fragments. And that's important because we want to understand not only the nature of genes, but also their distribution. And so just doing a little bit of sequencing allowed us to get a very, very nice look into this unstudied genome. So for the students, we set up a genome project BLAST website with all the data and annotations and so on. They were able to go through this and ask whatever questions they wanted. So we use something called Evidence Modeler, which is an ab initio gene prediction method combined with about 4 billion base pairs of mRNAseq transcriptome data. Out of that, we predicted 8,590 exons, very low number of introns, and about 8,355 gene models. So we have, out of all of this bioinformatics, about a 20 million base pair genome that encodes about 8,355 genes. Very dense gene packing, one gene every 2.3 or 4 kilobases. So really nice genome if you're a genome biologist because everything is tight, very few introns. It's kind of like the enlarged bacterial genome. So one of the first things you do when you have what we hope is a high quality genome data is you run what's called a BLAST-P search, where you take all the predicted proteins that came out of those gene models and you search against, in our case we have a local database which includes all of RefSeq, which is all the unique data at NCBI, plus all of the ESP data at JGI and so on. You figure out what the evolutionary signals are within the genome, and as you would expect, the largest number of hits for the BLAST-P are to red algae, green algae, and strominophiles, which are, I'll show you what that means in a second. But you also find that database size matters, that the large databases, for example, of animals and fungi, also pick up lots of hits. That's only because there's lots of proteins there, and their overrepresentation leads to them coming up as one of the top types of hits. The important thing is there's no big peak down here of bacteria or viruses or something else. So it suggests we have a pretty clean genome. All right, so we take all of that, those protein models for which we have confidence we have a pretty clean genome with this hit distribution. We then apply them to a phylogenomic pipeline where we feed each of the proteins into a pipeline to generate alignments and maximum likelihood trees. Then we look through those trees and say, what is the overall phylogenetic signature within the Porphyridium genome? And this process results in about 6,000 RAXML bootstrap trees. And using 2 different cutoffs of bootstrap, which is support for groupings at 90 and 70%, these are the groupings of the Porphyridium genes with things within the plantae. And this huge group shown in brown here are all members of chromalvulates. What does that mean? What it suggests is that the red algae clearly have genes that are shared with other green algae, plants, and glaucophytes. What it suggests is that it shares a huge number of genes with chromalbulates, and about half of its genome you can find in a chromalbulate. Does that mean chromalbulates and reds are monophyletic? No, it means that red algae are sources of foreign genes that have been incorporated in chromalbulate genomes. And it's really phenomenal. So the red algae have essentially littered the tree of life with their genes through horizontal and endosymbiotic gene transfer, which makes them one of the most important eukaryotes without really being known for that. Okay? So let me just show you what this gene sharing, if that's a weird word to you, what does that really mean? Here, for example, is what we call a plantae-specific gene. This is a gene involved, a protein involved in alcohol fermentation. It has a bacterial origin. It's found in the glaucophyte red and green lineage. There's not much support here, but it appears that of all the data that we have in our database, only the plantae share this bacterial gene. Looks like the ancestor likely gathered this gene from a bacterial source. So they call that a plantae-specific gene. What's a shared gene? A shared gene is this well-known gene, fructose 1,6-bisphosphate. Phosphatase is involved in cytosolic as well as Calvin cycle processes in the plastid. We had an ancient gene for the cytosolic version here. There was a gene duplication, and this plastid targeted version involved in the Calvin cycle only shared by the plantae, but the plantae share that gene chromalbulates through secondary endosymbiosis. And this is what I mean when I say the reds are masters of gene sharing. It's that many of their genes are sitting inside of chromalbulates like diatoms and dinoflagellates and haptophytes through the endosymbiotic gene transfer process. So the red algae have littered their genes throughout many, many of these lineages. So that's a automated analysis, and I thought just to let you know that things are actually a lot more complicated than what one can achieve with automated analysis. I will show you just sort of a manual look at some of the genome data. And so I chose here 10 contigs. This is the coverage. That means how many times any nucleotide in there has a read that supports it. It's really high coverage. So we have very, very strong genome data. Here are the numbers. They don't matter. This is about 4% of the genome here with those content. So these 10 contigs from Porphyridium encode about 300 trees from our XML set. What we did, we went through manually and we categorized those 303 trees in more complicated ways. And I don't want to go through all the complicated ways, but some of the ones are that the red algal gene has a bacterial origin, is shared with other plantae cyanobacteria, specific to plantae, and so on. So essentially, we tried to take, when you look at a phylogenetic tree, you can have a very, very complicated history, and you can't summarize that with one index based on a bioinformatic pipeline. So we actually looked at these 303 trees and categorized them based on these more subtle and more sort of complicated scenarios. So what did that tell us? The first thing it told us, if you look at the 10 contigs, that this sort of complicated scenario for gene origin and gene sharing is actually across all of the contigs. So each of those contigs represent different regions of the Porphyridium genome. Each of them shows aspects of bacterial origin, plantae specificity, and so on. So the genome is highly mosaic and chimeric across all those 10 contigs. So that's suggesting we have a very, very complicated history for that genome. So let's look in detail at one contig just to make this even more clear. So this is the famous contig 2035, at least to me. It's about 50,000 nucleotides. So there's all the gene models. They're lined up, and the arrow says the direction of transcription. Little spaces are introns. Lots of genes packed together like a bacterial genome. Very, very few introns and just a few of the genes. Okay, so that's a typical Porphyridium red algal genome contig. It's beautiful. All the stuff down here are the mRNAseq data that map to the transcripts. So they actually were used to provide support for the gene model as being real, because that predicted gene gave rise to a transcript that mapped exactly to it, and these coverage values show you that the transcript abundance across the contig. Okay, so relatively good-sized contig, lots of genes, good support for them being real based on mRNAseq, and having the characteristics that I've suggested previously, tightly packed, very few introns. So let's walk down that contig and see what the red alga tells us. So here's gene 35. Gene 35 is very nice. It suggests red-green monophyly, a eukaryotic gene with lots of other eukaryotes here. Lots of support for it using mRNAseq data. You would say every red algal gene should look like that. It should be a red lineage gene shared with other reds, sister to greens, and so on. So here's another gene, number 41. And that has a very different scenario, but this is unresolved. It's a protein kinase domain, so maybe this sort of function is spread throughout different types of proteins, so we have a complicated scenario. Eukaryotic protein, hard to interpret, lots of evidence. So we can call that unresolved and say, okay, some genes in the red algae we can't explain easily because they have these complex distribution. But here's one that's a little bit easier to explain. This is gene 17, and it shows a bacterial origin for a gene shared by the glaucophytes, reds, and the chromobulates. Again, strong support for it. So here's a gene that may have originated in the plantic common ancestor from a bacterial source, maybe a chlamydia source, and that was then probably passed on to chromobulates, not much support in the bootstraps. So that's already suggesting this red algal gene is bacterial origin has been shared then from bacteria over red algae to chromobulates as one potential scenario. So here's gene 28. In this case, the gene wasn't shared with chromobulates, but it's clearly of bacterial origin, but it's found in multiple red algae. Okay? So the same contig, another gene with a bacterial origin, strong support for that. That's gene 28. And finally, here's gene 4. And that is found only in Porphyridium in our existing database. I'm sure it's in other reds. We simply don't have the data yet. Likely a proteobacterial source. Is it a bacterial contaminant? Likely not. It contains introns. And it turns out it's sitting next to another gene, which is only shared by red and green algae. So again, very strong evidence for a recent bacterial gene transfer in red algae from a bacterial source, but not shared with other chromobulates. So looking at one contig from this photoautotrophic cell wall typical eukaryote provides a whole set of scenarios to explain their gene origin. Now the question becomes, we're in the process of annotating all of the transferred genes, trying to figure out what processes might underlie or why would selection favor this sort of gene turnover. And I point this out to you because for a long time people thought you had to be a phagotrope or a parasite to gather foreign genes. I'm providing you as straightforward evidence as I can to show you it's not the case. That single-cell eukaryotes are their own germline, and they can carry all kinds of foreign genes, and they can play fundamental roles in their evolution. All right, so from this just a really preliminary look, there's a lot of other stuff going on here. Some of the things we've learned thus far, the Porphyridium genome is readily assembled, very few introns, great model for doing genomics, functional genomics. We can study horizontal gene transfer, because it's very easy to get at those genes. We actually have 2 other strains of Porphyridium fully sequenced at 40x coverage. And this postdoc, Maria Matilda, is doing functional genomics, looking at how under different types of salt stress, how different genomes adapt to those stresses and identifying the genes that might underlie that. So Porphyridium is also very easy to cultivate, and it's a very good model for that sort of stuff. A number of people looking at lipids and starch. I won't talk about that, but all this work is also ongoing or it's already completed. The point I want to leave you with is that, contrary to what might have been thought for a long time, even good photoautotrophs with cell walls and no flagella can be sinks for genetic information, and they certainly are a source of genetic novelty, because they gave rise to the plastid in many chromobulates, and therefore all the nuclear genes that support the plastid moved with that. So they're, in fact, in a weird way, red algae have been ignored for a long time. It turns out that they're really impressive for their impact on the tree of life. All right. Okay, so people who study endosymbiosis are always looking for a better model, something that doesn't go back 1 billion years. And this is a really cool model I want to spend a few minutes talking about. So Pollyanella was discovered in 1895 by a German naturalist whose name was Robert Lauterbahn. And this is the model everyone has worked on until now, the plantae. I've talked about this, right? Red, green, glaucophyte, 1.5 billion or so years ago. Lauterborn described this, this weird Phylogenetic with what looked like little cells inside of cells, photosynthetic cells, inclusions inside of it, chromatophores, in 1895. And this organism was lost pretty much to history. There was one culture of it in Germany. When I was a postdoc in Cologne, I started working on it. And now we have many more strains. We rediscovered what Robert Lauterborn found in 1895, but now we recognize it as being a really, really amazing model. Why is that? Here are the plants that endosymbiosis, this one occurred over here. The pollinella is a completely unrelated origin. Is that fun? Or is it independent? If it goes away, I'll know the answer. You guys are tough on speakers here. Okay, so here we have an independent origin of an organelle. That's really exciting for people who study eukaryotic evolution. A few years ago, Wansu and others, I went back to my old phage library I made of the German strain here, and I was working with Melkonian, and I was able to recreate some of the plastic genome. This is the genome of the captured cyanobacterium. We were able to place the Pollinella chromatophora genome within a group called the alpha cyanobacteria. So we could say that organelle came from alpha cyanobacteria, where every other algal plant and plant plastid is also cyanobacterial, but nothing to do with this guys. An independent origin. So that was the first we were able to show by studying gene order and multi-gene phylogenetics. But I also want to point out one thing to you that many of you already know about, is that the alpha cyanobacteria are famous for being infected by cyanophage. And I'll explain to you why that comes into the story down the road. Very specific phage that infect this group here and are very famous and studied by a lot of people. Or not. All right. So I want to go through some what might seem more tedious molecular evolutionary nonsense, but it actually is about. It's fundamental to how we understand the evolution of genomes. And this is a young fellow here, Wan Chu, who's a postdoc in my lab. What can pollinella teach us about ancient events in genome reduction and plastic genome evolution? It's a long question, but what can we learn by looking at this model? We believe that pollinella endosymbiosis occurred about 60 million years ago, as opposed to 1.5 billion years. Okay. So what did we do? What did Juan do? First of all, we thought of a model. So he generated, looking at all the alpha cyanobacterial genome data that was available, he made a 268 concatenated protein tree, and he showed that the pollinella, there's the German strain I originally worked on, and another strain from Japan, we have others in-house as well. This is that 60 million year old endosymbiosis. This is the group that gave rise to the plastid in pollinella. Here is a Porphyridium group that also has reduced genomes. Okay? So genome reduction here, genome reduction there. Here is the genome size. Instead of being 3 megabases, maybe 1 megabase or 2 megabase. Okay? So that's what can we learn about genome reduction, and the GC content, as you can see, AT richness goes up as you reduce the genome, shown in both cases for endosymbiont and for naturally occurring strains. So the first thing we wanted, I said, one, prove to me that those are not the same group, that this is not actually derived from the same one. He did something called the AU test, which is a test used by molecular evolution types. And essentially what it shows is that uniting these reduced genomes leads to a very, very low probability. That's not the case. So we have 2 independent cases of genome reduction via endosymbiosis, via natural forces acting on free-living cyanobacteria, fecal cyanobacteria. So that's the first thing that you make clear. So genome reduction is characterized by massive gene loss. Why does that seem so obvious now? Thereby called genome reduction. And it's caused by the loss of paralogs. That is, if you have 2 copies of a gene, you tend to lose one and keep one. Okay, that's just a fact. So how did paralog deletion proceed in pollinella and the picocyanobacteria? Very similar. Here's the proportion of multi-copy genes over the total gene number, very low in our captured endosymbiont, low as well in the small genome Porphyrococcus, and different for the other Porphyrococcus and Synechococcus. And again, this heat map shows that many of the genes in pollinella and these guys are in single copy or in low copy. So genome reduction leads not only to GC content, difference, but it also leads to loss of paralogs. The story has an ending. Okay. So let's now focus on a really important aspect of genome evolution, that is the use of conserved genes for phylogenetic. So anyone who's done plastid gene phylogenies have used the photosystem proteins, D1, D2, PSBA, PSBB, PSBA, and so on. What can they tell us? So most photosynthetic eukaryotes in their plastid have a single PSBA gene. Pollinella, the more recent Endosymbiote, has 3 of them. Okay? So the question we ask is, what are the evolutionary histories of the 3 Pollinella PSBA genes? Here's the photosystem 2 with the D1 and D2 proteins as part of the core reaction center, really, really important components. And you would think highly conserved and fail-safe phylogenetic markers. So let's look at the inheritance of PSBA genes in Pollinella and Prochlorococcus. So how do you identify orthologs in genomes? We look at synteny. So there's actually 2 types of PSBA, the D11 and D12. They have somewhat different functions of light capture in cyanobacteria, and that's what those 1 and 2s mean. The important thing is, by looking at the 3 genes in our 2 Pollinellas, we can identify the synthetic orthologs in the cyanobacteria based on the neighboring genes. Okay? So there's been gene loss, but we can pretty much say this gene is related to the common gene that was in this ancestor, and so on. Okay, so we identified orthologs based on synthetic position, and that seemed to work pretty well. We noted that Pollinella only has the D12 type of PSBA. So given that, if you make a tree of those guys, orthologs should then line up region by region, region 1, region 2, region 3, because they maintain the vertical history through evolution, show it by the synthetic position. Surprise? Not at all that. In fact, what they do is that they line up based on the species they come from. That is, if you have 3 PSBA genes and you're a Pollinella strain from Germany, your 3 guys at the DNA sequence level are more closely related to each other than they are to the synthetically positioned other genes. How do you explain that? Anyone who studies molecular evolution will know how you explain that. That's called concerted evolution through gene conversion. Okay? So unambiguous evidence that in the context of other cyanobacteria, the Pollinella genes are in fact undergoing concerted evolution, where the synthetic position no longer reflects history, it's the species you're in. Now, does that matter? So plastid genes have undergone paralog deletion, starting with, for a lot of genes, 3 PSBA genes, most eukaryotes ended up with 1 in their plastid. Are some Pollinella genes in the process of being inactivated or deleted? Can we catch some of this evolution in action, going from 3 to 1, which is what we expect to find as an algae in plants. So without getting into very much detail at all, I can show you a tree in which we then had 7 different isolates from different countries. Again, we use single-cell genomics. I won't talk about that for a few more minutes, but we were able to easily isolate the PSBA genes from the Pollinella strains from different continents. And in fact, we found 1 clade shared of the same region across different areas, all undergoing deletions in their PSBA genes. So they were actually in the process of losing the region 2 gene. So this was, again, suggesting they were headed to the 1 gene solution that was found in algal plant and plastids. So do other genes, duplicated genes, have an unexpectedly complex evolutionary history? Yeah, of course. That's why I'm talking about it. Turns out that the photosystem proteins do the same thing. Here's PSBD. Here's 2 synthetic regions. Again, the trees line up based on species, not based on region. Okay? So what this is telling us is we have, and this is the expected tree, is that concerted evolution acts on even these highly conserved proteins in unpredictable ways. So implications in the PSBA case in Pollinella reflects what occurred 1 billion years ago on the plastid of the plantae. The single copy genes might be the result of independent homogenization and random deletion. If this happened after the split of the plantae lineages, that the reds, greens, and glaucophytes carried 3 genes and then underwent independent deletion and conversions, this tree is not going to reflect the tree of the organelles. It will reflect this process of molecular evolution. And this could mislead deep phylogeny reconstruction. The same is true for other genes like PSBD, E, and C that I didn't go through here. What does that mean for people trying to reconstruct the history of plastics? What does it mean for the phylogenetic signal? So what I asked Juan to do then is to take all of the proteins that have this complex signal, make a phylogeny with that, take all the proteins that were single copy or in the cyanobacteria, or are duplicated genes but had a single copy for millions and millions of years, we believe to be good orthologs. When he did that, here's what the complex protein scenario showed. Incredibly strong support for the glaucophyte, red monophyly, and here are the greens. This is all those paralog deletion conversion types. We took what we thought would be the good molecular markers. You got a completely different picture. You got the monophyly of reds and greens and the glaucophytes at the base with strong bootstrap support. Okay, so that seems like a subtle notion. Who really cares? But in fact, if paralog deletion and conversion are fundamental roles of gene families, and all of deep evolution has a big problem, which you can't account for it, depending on when the process occurred, it can mislead phylogenies significantly. So I like this tree. There's always a tree one likes. I like this tree because it puts the glaucophytes as the first divergence, and this has been the hypothesis for many years because they have unique features like the peptidoglycan wall between the two membranes. They have not proboscisomes, they have carboxysome-like structures. They're much more cyanobacterial in their appearance and in the pigmentation. So this scenario is a little bit harder to explain, but it turns out that this conflict here may, in fact, be explained by something you could never have figured out until you looked at Pollinella and saw that the process that actually defined these genes. So I've explained all of that here, so on and so on. More generally, our results point out that caution needs to be exercised when choosing serially duplicated genes to resolve deep phylogeny. And that's not only for blastids, but for any deep phylogeny for eukaryotes. That stuff is in a paper that just came out in Molecular Biology and Evolution in much more detail if you're interested. Okay, so that's it. Now let me move on to a little bit about single-cell genomics, the time that remains, pending thunder. So we've completed a genome project of Pollinella, and I'll give you one slide that sort of summarizes the absolute bottom line. Let's say the Plantae and Pollinella both got cyanobacterial endosymbionts with about 2,500 genes. Here's what our genome project for one of those is a Korean strain of Pollinella we have. What is the real bottom line? This is what the Pollinella plastid looks like. About 850 proteins, 1,000 to 2,000 lost, 42 transferred to the nucleus. Compare that to famous arrow, to algae and plants. The genomes are 80 to 200 proteins, 600 to 1,000 in the nucleus, as I was suggesting, the same number being lost. So this is presumably the direction of evolution. I was showing with the PSBA with paralog deletion. Ultimately, we're going to have the genome reduced in a few 100 million years maybe to something like in plants. And we'll have many of these genes moved to the nucleus through endosomatic gene transfer. So that's why Pollinella is a really cool model to understand how this stuff happens. Okay, so those are really great and exciting steps forward for my lab, but can we take any of this to Mother Nature? So single-cell genomics is this here. It's pretty straightforward. You use some sort of a sorting, fluid or a flow cytometer. You isolate single cells marked with either a dye or based on size. You amplify the DNA from a single cell using whole genome amplification. And you sequence that pool and reconstruct the genome of that single cell, of genomics at the single cell level. It's very, very exciting. At least we think so. And the initial work we've done has been done with the single-cell genome sort of isolation folks from Lunas and Mike Zaraki when Wansu was there at the Bigelow lab. Now we're doing it on our own. But essentially what you do is you get natural environment sample, you isolate it using flow cytometry, you capture them in microtiter plates, lyse the cells, amplify the cells, do rDNA amplification and sequencing to find out what these cells actually were, then you go on and do whatever you want. Okay? So these methods were developed for bacteria, Roger Laskin and other folks at the Venter Institute, and proving that contamination was minimal was critical. About 0.1 to 10 picoliters of medium is transferred to each cell. That's always a concern. What my lab did is we took these methods developed from bacteria and viruses and applied it to eukaryotes, to where we have 100 million base pair genomes instead of 2 to 5 million base pair genomes. Okay? So the initial paper that did this was published in Science in 2011. I'm going to talk about a follow-up study we did, which relates directly to endosymbiosis. So first thing we can ask is, what do you expect to find when you capture single cells in the ocean? Do we expect to find an exilian culture with high-dense algal concentrations? Or we expect to find this famous picture from Recruitment showing diatoms and bacteria and viruses? This is what we know what the nearshore ocean environment looks like. Therefore, there's no reason to believe that if you captured a single cell from here, that it would look a whole lot like a single cell captured from there, because they're living in very different environments. The question is, what is the difference there? So now that you understand the Pollinella model perfectly, hopefully, let me now introduce you to Pollinella's sister, also called Pollinella, but now called ovalis. It's a great paper by Johnson et al. In 1988. They showed that this amoeba's sister to Pollinella chromatophora eats cyanobacteria, which were found inside in a vacuum in its cell. So we use this model to test the connection between what you eat, horizontal gene transfer, and symbiosis. So Nicole Poulton at the Bigelow Labs, she ran the flow cytometry. We chose small-sized cells that didn't have chlorophyll autofluorescence. They were marked with lysotracker, and we took the small-sized cells over here. So thousands of these cells were sorted. Their DNAs were lysed in picotiter plates, rDNA amplified, sequenced, and then a catalog made of what each cell represented. From this, we were able to identify a number of cells that were rDNAs very similar to Pollinella ovalis. That's we were able to get a hold of these cells from the ocean, in this case off the dock in Chesapeake Bay. So here is the photosynthetic Pollinella, here is ovalis. We looked at 6 cells that came from here, which the rDNA sequence showed clearly was most closely related sister so far to the photosynthetic cells. So after a lot of bioinformatics I won't talk about, we ran the same blast analysis. Now instead of finding, remember that nice decay pattern of plants, animals, fungi with no bacteria, well, here's what it looks like when you go to the real world. Here you have a large number of bacteroidetes, viruses, proteobacteria. Okay? You still have the top hits to animals and so on, brown algae, the big database stuff, but you're finding that each cell has its own unique blend of DNAs associated with it. We described this when we wrote that paper on picobilliphites, the first time we ever did this, and we postulated this is because they're eating different things. The same thing seems to be the case in Pollinella ovalis, a phagotroph that has different DNA abundances in different cells. So if you take those genome contigs and you analyze them gene by gene, here, for example, is 11,500 nucleotide contig that is clearly cyanobacterial derived. You find, in fact, that they are eating what appear to be alpha cyanobacteria. So the hits for these proteins are all alpha cyanobacterial. And here's one for a long conserved protein called porin. Here is the DNA recovered from one of the cells, and its sister to alpha cyanobacteria. Just remember that the plastid in chromatophora is of alpha cyanobacterial origin. On top of that, we find some large contigs, I'll talk about it in more detail in a second, that encode these D2 proteins of cyanophage origin, which are nested within alpha cyanobacteria. So we're getting a signature in a large piece of DNA, not only of cyanobacteria, likely as food, but cyanophage that are likely infecting the cyanobacterial food. That's one explanation for what this data means. Can this sort of behavior actually lead to horizontal gene transfer? So we're developing much better methods to do this right now, but this is the first time we tried it. We're able to find one really great example of this. Here is a piece of the Pollinella ovalis nuclear genome, 3 genes with introns, that's what those guys are. Coverage, lots of DNA coverage, so we don't have mRNAseq data because it's a single cell, but we have coverage. Eukaryotic gene, sitting in the middle is a diaminopimylate epimerase of alpha cyanobacterial origin, right there. This is a protein kinase of eukaryotic origin. So we not only find signatures of alpha cyanobacterial food, presumably in the vacuoles, we actually find evidence, direct evidence of a eukaryotic gene of alpha cyanobacterial origin in the nuclear genome of Pollinella ovalis. The same gene is found in the plastid genome of the chromatophora, its photosynthetic sister. That's the first time we were able to connect food, the horizontal gene transfer to endosymbiosis. To put that as bluntly as I can, amoebal, food. That's the model. So we spent a lot of time worrying, is this real and so on, and I'll just show you that this region of the DNA of high interest has lots of sequence coverage. There's some repeated areas that have low coverage. We found evidence that we can span those regions in some reads. On top of that, we found that we can map, from all 6 cells, we can map either incomplete or most of that same region to that same piece of DNA, suggesting that we in fact have an intact genome fragment that's present in all the cells. We did PCR across these and also found the gene, so we believe it's a bona fide horizontal gene transfer. So that experiment allowed us to connect phagotrophy to horizontal gene transfer of the same food source that gives rise to the plastid in the transition from a phagotroph to a phototroph in organelle. So these methods, we believe, are really powerful. If the DNAs are involved, are different enough, we can actually distinguish the genome of the nucleus of the host from the genome of the food, the alpha cyanobacteria in this case, from the genome of the cyanophage infecting the alpha cyanobacteria. We dug a little deeper in the cyanophage picture. We were able to find 19 contigs, which total about 210,000 base pairs, on which we believe we have captured most of the cyanophage genome. And that's shown here. So we don't have a reference genome, not a mapping procedure, a de novo assembly of the cyanophage from fragmented single-cell genome data. So those 19 contigs are shown here. 4 of those contigs encode about 100,000 base pairs, about half the genome on 4 contigs. And here's showing the growth in the genome, the fragmented genome size as we add contigs here. Not only that, if you look at the proteins encoded on here, the 179 proteins, we find clear evidence of similarities to Synechococcus cyanophage SSM2, Prochlorococcus SSM2, and other cyanophages as well. So we're able to identify what the likely source of the cyanophage fragments are. I should add that by looking at the contigs, all of the cyanophage contigs are unique, suggesting we don't have populations of different cyanophages inside the cell. So it looks like a single genome that is being partially reconstructed here. And if you take 5 conserved cyanophage proteins here and make a tree, you in fact are able to show that P. ovalis-like cell 1 cyanophage is sister to Synechococcus cyanophage SSM2 and Prochlorococcus in this phylogenetic analysis. So we believe we're able to capture much of the cyanophage genome on fragments. We're able to identify that cyanophage inside of the bacteria, inside of the roots. All right, so a lot is being done, can be done. There's a lot of, we're developing network methods to study single-cell genome data. There are lots of things we can do. One final point I want to leave you with is that as much as this stuff is powerful, the interpretation of algal evolution is really, really complicated. And the Porphyridium data should make clear that even a typical red alga can have a very, very complex evolutionary history. I'll show you one last tree that will drive this point home. Here we looked at genes that are shared between photosynthetic Pollyanella and phagotrophic Pollyanella. These data blur the line between anciently heterotrophic versus phototrophic lineages. This is a ribonuclease Z gene involved in tRNA maturation, cyanobacterial gene found in a chromalbulate, some picorbazenophyites, and the photosynthetic and the non-photosynthetic Pollyanella. How would you possibly interpret this tree? Does this suggest that these guys were actually anciently photosynthetic and are retaining a cyanobacterial gene shared with the plant, and that was transferred somehow to the algulates? Is this the chromalbulate rhizaria group here next to the red? I have no idea, but this is exactly what you find. So when you say that, when I say that Ovalis is anciently heterotrophic, it's a little complicated because in fact carries cyanobacterial genes shared with photosynthetic organisms. So this whole notion about, one can say that animals and fungi were likely never photosynthetic, no problem. You get to the protists, it's much, much more complicated. And we don't know exactly what this means. Ancient cyanobacterial gene transfer, loss in other lineages, we don't know, but it makes for very, very interesting but complex scenarios. And here's the intron distribution showing they're real. Okay. So I'm going to stop there, and I'll be happy to take questions before I end. I want to thank the great people in my lab, who've done fantastic work. And I want to thank the folks who fund us. And of course, I'm going to thank our dog for hurting us for this picture. And thank you for your attention.
So, questions?
Yeah.
So, given the plasticity among bacteria and their ability to modify their metabolism depending on changes in, even minor changes in their, microheterogeneity in the environment, why wouldn't you expect that in some of those microalgal groups to?
Yeah, I think that would be one of the prevailing scenarios, is that if you're small and you're germline, you can capture genes. People have simply thought that based on the fact that they have a nucleus and they often have sex. I mean, bacteria do these things because they don't have access to normal meiotic sex. They have other routes. If you're a sexual outbreeding protist, why should you be like a bacterium? And you have a nucleus that should protect you from gene transfer. You expect viruses, probably. We know there's lots of viruses that have been described, or I'm sure do infect a lot of these same cells. Yeah, I think that's one of the scenarios. I think is that, but really, if you were to say that to a room full of eukaryotic biologists, that Porphyridium is simply a big genome bacterium, in spite of its cell wall, in spite of its nucleus, in spite of its eukaryotic, in spite of all the things that make it eukaryotic, people would say, well, that's surprising. But I think that's what you're saying is probably the answer, is that if you're a small cell that lives in the ocean, that you're actually transferring the gene pool amongst your genome in the way that bacteria do. It's just nobody ever expected that to really be the case. They really thought that eukaryotes had this wall, and that they have other ways of generating genetic diversity through gene duplications, and so on, sex, outbreeding. They didn't have to revert to this much more dangerous game of capturing foreign genes and turning them on and so on. But it looks like it's exactly what they do. But I should also say that this is going to differ. If you're a multicellular organism, you're not going to have this problem. Having sequenced the sea slug genome and looking for horizontal gene transfer from the alga, there ain't none. So I can tell you that just because this scenario will fit some lineages and it won't fit others. If you're a multicellular organism with a germline, where there's some physical separation from the food, and it's not going to be the case. So this is it's going to be a much more complicated scenario. But I do think Porphyridium is, as you say, like just a big bacterium. Yeah.
I'm interested in horizontal gene transfer. Do you have already an understanding how the organism or the cell decides which genes to incorporate into the nucleus and which to leave at the plastid or the organelle?
I presume it's all selection. I mean, there's no reason to believe that there would be any. I think they sample. So I think the more subtle question is that, well, at least the way we think about it, there's a little glimpse into the connection between what you eat and endosymbiosis. That's the first time we've ever seen that connection. So that suggests to us is that the consequence of a long-term phagotropic lifestyle, especially if you focus on a very abundant food source like alpha-cyanobacteria, could ultimately lead to some rather fundamental changes in your lineage. And the point is that our cyanobacterial gene transfers pre-adaptation. I mean, they do a job, but once does the cell already have the capacity to somehow fiddle with that captured cyanobacterium before it becomes a plastid? I think that's what we're thinking, is that if you have a long enough time frame and a particular food source long enough, likely there's not only chance for horizontal gene transfer, but also for more fundamental properties to change. And I didn't talk at all about the other big gene transfer story, which is a lot of green algal genes found in chromalgulates. The P group xenophytes, as you probably know, were also very, very dominant in the oceans, and they still are. So a lot of genes from them have ended up. The question everyone asks is, was there a cryptic endosymbiosis in chromalgulates which drove the gene transfer or not? So I think it's a combination of a particular food source that is a ratchet for gene transfer, whether that ratchet simply ends up in giving your genes lots of novel genes, or something fundamental like endosymbiosis or kleptoplasty, any sort of interaction beyond food. I think that's what is really interesting, and I believe any other gene transfer is just selection. If the gene is transcribed, translated, and provides some reasonable function that's not too deleterious or positive, I think it'll probably spread. Yeah.
I have two questions.
Sure.
It's a follow-up to the question I had earlier.
Yeah.
And I think it's follow-up similar to their questions also. In bacteria, for example, the gene transfer, gene transfer should happen in gene cassettes,
Right.
not the single genes, but as groups of genes, and normally with transposons or
Right.
some signals to facilitate this gene transfer. How about here? You mentioned that in plastids there's no transposons, and in the sample you gave us, it was an amoeba. And
Right. There's no evidence there.
Single genes.
So we look for those sorts of things. I mean, we try to imagine, is there a virus, is there a viral vector? We find evidence of any sort of viral sequences within the genome that suggest that there's a medium, is a transposon that's shared between the food and the, those are some of the things we look at. Now, in terms of the physical movement of DNA, that's been shown to happen by lots of people experimentally. I mentioned today, someone was talking to that Ralph Balk designed these eukaryotic genes inside the plastid of Arabidopsis and selected for them to be found gene trans. So I think physically DNA can move between organelles, as long as the organelle's death doesn't kill the organism. If it has multiple genomes, multiple plastids, you can, when one lyses, the DNA can integrate. And then, so we looked at, now in terms of the physical, the synteny of cells, we never found an example of recent gene transfers with a number of cyanobacterial genes or any genes side by side. We addressed that question for these green genes we saw in this diatom paper in 2009 in Science. We looked at across all the diatom genomes and used this broken stick test to figure out what the size, what the intervals between the gene transfers were, and it was random. So my feeling is these things come in. If they come in as a cassette or as a single sort of large region, they get broken up over time. We have not yet found that perfect smoking gun of transposons, bunch of bacterial genes, and then thinking. So ultimately, you can't, unless you have a polycystronic message, you're not going to express those genes anyway. So I think in terms of the eukaryote's point of view, moving them around, getting them into their own regulatory regions is probably what they want anyway. So I don't think that this bacterial model would last in eukaryotes, but we've never found that particular thing you're talking about. No.
You have another question?
Yeah, go for it.
About the phage genome that you said you identified, the specific to phages. I have some idea about phages, and most of the time when we get the phage genome, there's always a lot of, maybe up to high percentage of host genome, very similar to the host genome.
Yeah.
So, but in that case, you were not able to identify them there.
No, so what we did in this case is that we had 19 contigs, and so I didn't show the network analysis, which showed the highly modular nature of that protein. That was one slide too much. We actually looked at the distribution of, and those cynophage proteins and genes are distributed amongst a bunch of different cynophages. They are a highly modular genome with lots of gene transfer. So there's no doubt. We didn't find any host eukaryotic genes in any of those. Now, if there is a small fragment and it doesn't have cynophage genes next to a host gene, we'll never be able to identify it. But we looked for larger contigs that have cynophage, and all the ones we found didn't carry any SNPs, so suggesting it was a single genotype. And secondly, when we looked at the distribution across all, we used 17 sequence genomes, we saw lots of sharing, suggesting that this cynophage, like every other one, is a member of a common gene pool. And it has, most of its similarity is to this SSM2 type cynophage, but it actually has lots of other evolutionary histories as well. And that was pretty normal compared to what we expected.
Thank you.
Yeah, you're welcome.
So, what family of genes are lost from endosymbionts that cause them to lose their independence and become plastids or mitochondria?
Yeah, so they tend to lose tRNA synthesis genes. They tend to lose transporters. So if they're into harvesting any sorts of sort of nutrients or even ATP in some cases or nitrates, those all tend to be lost. So the Pollyanella genome. So that's a big mystery as to how all of this process occurs, and we're generating a proteome from the Pollyanella endosymbiont, trying to figure out these membranes, what proteins are there. There's a lot of controversy about how you turn that endosymbiont into a functioning machine. Does the host do everything, which is what we tend to believe, or most of everything, or does the cyanobacterial endosymbiont contribute a large part of the machinery? But the things that it definitely loses are its transporters, and tRNA synthetase, and in this case we found some of the photosystem genes transferred to the not the major ones, but the smaller subunits, which we believe are really kind of like anchors to control photosynthesis, so they import those to control function. These cells are their division is perfectly aligned with the whole cell division. So yeah, we haven't gone further than that. In fact, the 41 genes we and others have found in the nucleus of Polynella don't give a lot of clues as to exactly how that works. A lot of the proteins are highlight intensity proteins. So the first thing we realized, we weren't that smart, we realized that, well, you get a porcelain organelle, how great. It turns out the worst thing you can have is a porcelain organelle that soaks up light, and you have to, so there's a whole bunch of highlight intensity proteins that have moved to the nucleus and have amplified into many, many genes. So that's one of the first outcomes that happen. The actual workings of division and control of the endosymbiont are likely host proteins. We'll have to, I think we'll only identify as if we sequence the protein of the organelle and are able to figure out what parts of machinery come from the endosymbiont, what parts come from the host.
Any more questions? Well, in that case, I'd like to thank our speaker.